we will discuss role of a restriction engine, types of restriction engine, what is their function in RDT, why they are called major tools in RDT. So next we will start how we can give the particular name of restriction engine. There are so many RE restriction engine are discussed today. Near about more than 900 RE is discovered till date from more than 230 bacterial strains. Very important questions. Bacterial strain. Different kind of bacterial strain are used. About more than 200, near about 230 bacterial strains. Then what is the total figure? That is called more than 900 RE is discovered today. So one mass question and MCQs may be set up from this one because this data is given in a NCERT book. So let me discuss how this particular name is given. What are the rules which are used? How we can give a particular name to the restriction endonuclease engine. For example, we will take the example of a EcoR1 restriction endonuclease engine. Here some words are written. For example, the first rule. What does it say? The first alphabet letter E. It is come from the name of bacteria. The bacteria which is called Escherichia. It means this is called genus name. Genus name. And a genus, a generic name is Escherichia. It means when we return a particular name to a particular restriction engine, the first letter indicate which is called E. It is taken from the genus, the generic name of that bacteria. Or here the genus name is called Escherichia. So what we will take? We take capital E, like binomial nomenclature of biological classifications. Second rule, here two words are given which is called CO. Both are in small letter. It means it is, what is this? This is called species name, specific name. Or how you can write the specific name? COLI, coli. So what you will take? We will take two alphabet letter C and O. So we have pick up from here or we write this one. It means this is the scientific name of a bacteria whose name is called Escherichia. So what will we take? We take three alphabet letter. The first is the capital. This indicates it is the genus name, generic name. The second two letter CO, both are small letter. It is taken from the next name. Next name is called species name. So right here COLI, coli. So we take CO. Okay. The next word which is given here is called R. R indicate strain of bacteria. It indicates the strain. It means type. The complete name is called RY. This is called RY. So we will take only R. So this is the strain of bacteria. R we can put here. The fourth rule is in Roman. This is figure is given, which is called first. This is called Roman letter. So this Roman letter indicates order of discovery of RE. Whether it is a first order, whether it is a second and the third order. In biotechnology, there are three types of RE discovered. Type 1, yeah, you can say fast order. Type 2, write in Roman. Type 3, this is called third order. So it means it is the first order of enzyme which is discovered, which is extracted from the bacterial cell, the bacterial cell which is called Escherichia coli. So these are the some points, these are the rules how we can be write the scientific name of restriction enzyme, because to avoid the puzzling state there are so many different kind of R is discovered. Near about this figure is discovered from this total number of bacterial strain. So that's why a standard naming is required to give the particular name to a particular restriction endonuclease enzyme to avoid the confusion. So here from this section or this topic, the question may be asked from the 3 marks. So very simple, like the binomial nomenclature, there is no confusion. Write the example of echo R1, E indicate Escherichia, this is called genus name, generic name, CO indicate is the species name, and the R indicate what kind of strain. This is called RY, only write R, and in the Roman 1 indicate the letter of discovery of enzyme, whether it is the first order, second as well as third. So this is the topic about the nomenclature of restriction enzyme. Next one here, one marks question that may be asked from next one, that is called palindromic DNA. Palindromic DNA, or you can say that palindromic nucleotide sequence. One marks question that may be set up from the definition point of view. What are palindromic nucleotide sequence? Very simple definition. A group of letters which is written in a word which can give the same pronunciation when it is read forward as well as backward. This kind of group of letters is called palindromic sequence. If this group of letters which is written here, it is behaving like as a nucleotide base pair, this kind of DNA is called palindromic DNA. For example, take a simple word, madam, M-A-D-A-M. Right here, read here, madam, M-A-D-M, backward M-A-D-A-M, madam, same pronunciation. Like in Hindi, write महम, इंग्लिश में लिख सकते हैं M-A-H-A-M, don't write E, महम, M-A-H-M, बैक रीड करें M-A-H-A-M, महम. Malayalam, one more word which is given in NCERT, Malayalam, M-A-L-A-Y-A-L-A-M. Read in forward direction, read in a backward direction. If these alphabet letter you can be assumed, you can be considered, suppose this is called adenine, you know very well, adenine is the nucleotide base pair. It is coming to the purine one. If this word is read in forward direction as well as the backward direction, orientation is same. It means the reading plane is the same. So this is called palindromic nucleotide sequence. Any sequence of letter which can be read in forward as well as a backward direction, which can give the same reading frame, which can give the same pronunciation. So this is one more example. You can take the nitin, N-I-T-I-N, read forward, N-I-T-N, backward, N-I-T-I-N, nitin. So these are the some examples which represent the nucleotide sequence. You can be take the palindromic DNA. Take the double stranded DNA 5 dash to 3 dash G A T T C फॉरवर्ड हो गया. इन बैकवर्ड डायरेक्शंस 5 अपोजिट 3, इधर अपोजिट क्या है 5, तो दिस इज कॉल्ड फॉरवर्ड, दिस इज कॉल्ड अप. ये 5 प्राइम टू 3, तो G A T C अपोजिट क्या बन जाएगा G A T मैच करेगा A मैच करेगा T, अगेन A मैच करेगा क्या है T से, और C मैच करेगा C. डेट अगर 5 डायरेक्शन से 3 पढ़ते हैं तो यहाँ पर फर्स्ट क्या है G A A T C. इसी को फॉरवर्ड की जगह पर बैकवर्ड पढ़ते हैं. जैसे यहाँ फर्स्ट से G आया था, यहाँ पर G, फिर आगे क्या है A आगे, फिर वहाँ T T आया था T T, और नेक्स्ट जैसे यहाँ C आया था तो यहाँ पर क्या है C. तो such kind of double stranded DNA which contain nucleotide base pair which can give the same reading frame when it can be read in forward direction as well as the backward direction. So this kind of DNA is called palindromic DNA, or such kind of sequence of nucleotide base pair is called palindromic sequence या palindromic nucleotide sequence. तो these are the some example. You can be take the word Malayalam, you can be take the word Mehem, you can take the word Nitin, you can be take the word of madam. So write in forward and write in backward. You can be read in forward and backward. So both it can be give the same pronunciation. So these are the some example which are used here. Next we can study steps involved. Steps involved in formation of rDNA. Formation of a rDNA. rDNA means recombinant DNA. With the help of RE, restriction endonuclease enzyme. So we will take, we will draw a flow charts how recombinant DNA is formed with the help of a RE. So these are steps. Suppose this is called foreign DNA. This is called vector DNA. Vector DNA. You can be right here and here. No matter. It will be no change. Here write double stranded DNA है GAATTC. इसका कॉम्प्लीमेंट्री क्या बनेगा? इस के अपोजिट में क्या है C, यहाँ पर क्या है T, अगेन T, T मैच करेगा A से, और अगेन T मैच करेगा क्या है A से, और C मैच करेगा क्या है G से. सेम यहाँ पर सीक्वेंस वी कैन यूज GAATTC. इसका अपोजिट मैच कर दीजिए CTTAA and G. So try to make the cut here. This sequence with the help of a RE. With the help of a RE, we have to remove this sequence. We have to remove this sequence in the same manner. We also have remove upper, which is removed, that is come here. Which one? A, T, T, or C and a G. When these two sequences they are come close to each other, they are complementary to each other, what they will be make? This portion will become here. That is fit into this portion. How it will be again become double standing? G मैच करेगा C के साथ. इन लोअर पोर्शन में क्या है? T, T, A, A. ये सीक्वेंस क्लोज आएगा और यू पेयर इन बेस पेयरिंग रूल फॉलो करेगा. यहाँ पर T है, अपर में क्या है? A. यहाँ पर T है, अपर क्या है? A. यहाँ पर A है, एडेनिन है, वहाँ पर क्या है? थाइमिन. एडेनिन है और थाइमिन. तो it can be start to match in this way. In short, we can say that there is the alignment of two. One is called foreign DNA segment, another is called vector DNA segment, and these two segments they again form the double stranding DNA. It means here the combination of two fragments are taken. One it is taken from the foreign DNA, another is taken from the vector DNA. This kind of DNA is called rDNA. R means recombinant DNA. With the help of RE, restriction endonuclease enzyme, because these restriction endonuclease enzyme they can make the cut at the particular site. They can be identify a particular specific sequence. That's why they can be made. They can be cut the foreign DNA, they can make the cut the vector DNA, and these two single standing hanging strand they can be so again complementary to each other. They show the pairing with each other. They can be again combined to each other. They can be form the hydrogen bonding between their respective nucleotide base pair, and again two different types of DNA is combined to each other, and it can be formed a new copy, which is the mixing of two copies, foreign DNA as well as a vector DNA. So this is called recombinant DNA. In short, recombinant DNA is also known as rDNA. It is equal to vector DNA and foreign DNA. Foreign DNA plus में vector DNA. This is a very simple definition of rDNA, recombinant DNA. ठीक है, combination of foreign DNA plus में vector DNA is called rDNA. With the help of enzyme, which is called restriction enzyme, because these enzymes make the cut at the particular site. That's why these single stranding sticky end they are combined to each other, and they can be complementary to each other. This is the way.